Hello and welcome to today's Saturday session, where our topic for today is cell structure and continuity for Leaving Cert Biology. So throughout today's session, we're going to be looking at the structure of the cell, all of the different components of the cell within animal and plant cells, and then we're going to go on and look at how cells can actually divide through mitosis and meiosis to produce new daughter cells. So when we talk about the cell is the smallest unit of matter that can carry on all of the processes of life. So you might remember from junior cycle, there's a number of different characteristics of life that a cell or something would have to have in order for it to be classified as living. So some of those would have been movement, respiration, sensitivity, growth, response, excretion, and nutrition. So cells are capable of carrying out all of those processes of life. So all living things are composed of one or more cells. If it's composed of one cell, it's known as unicellular. But if it's more than one cell, it's referred to as multicellular. So we will be an example of a multicellular organism. Cells are the basic units of structure and function in an organism, and they can only come from the reproduction of existing cells, and we will be having a look at that towards the end of the session. There's two types of cell division which we'll be looking at. We have meiosis, and then mitosis will be the main one that we're going to be focusing on today. So cells contain a variety of internal structures referred to as organelles. So an organelle is a cell component that performs specific functions for the cell. And each of the individual organelles we look at in the next couple of minutes, we're going to refer to what their individual functions are. So some of the organelles we're going to be looking at would include the cell membrane, the nucleus, the mitochondria, chloroplast, ribosomes, the cell wall, and the vacuole. Some of those are found in both animal and plant cells. Some of them are only found in plant cells only, but we will get to that in a couple of minutes. So in order to be able to view the different cells and see what the different organelles are, we have to use a structure known as a microscope. Now, the microscope you would have used throughout your experiments as part of 5th year and 6th year, if you get that far, is the light microscope. Okay, so there's a light source down here at the bottom, which is going to provide some light for you to actually be able to see the specimens which you're examining up here. So a couple of different parts of the microscope. We have our lenses. So we have our objective lens. Normally there's 3 of them there. You would have by 4, by 10, and by 40. You have your eyepiece up here, which is usually by 10, so it magnifies things 10 times. You have your stage. This is where you're going to push your slide. You have your clips, which are going to hold this slide in place. You have your diaphragm. This is this structure here, so you can open and close that. That's going to control the amount of light that's actually getting to the specimen. You have 2 focus wheels. You have your coarse focus wheel. That's to focus the image, and then the fine focus wheel is for focusing the image as well, but that's more for precisely focusing the image. And then the light is for what's allowed for the specimen to be viewed. So just a quick note on magnification. So the total magnification is the magnification of the eyepiece multiplied by the magnification of whichever lens of the objective lens you're actually using is. So the eyepiece is normally 10 times on the light microscope you'll be using in the lab. The objective lens is either going to be 4 or 10 or 40. So in this case, if you're looking through the eyepiece, which magnifies it 10 times, and then the objective lens magnifies it 4 times, the total magnification is 40 times, because it would be 10 by 4, which is 40. Now, if you change that objective lens, so if the eyepiece was still 10 times, because that can't change, but the objective lens changed to 40 times, so you're using the highest power lens that the microscope had, then you would multiply 10 by 40, which would give you 400 times magnification. So that means that what you are viewing, or what you can actually see as you look into the microscope, is 400 times bigger than it actually is down on the slide. So we're going to start off with the animal cell, and the animal cells will contain the following structures. So we have our cell membrane, we have our ribosome, our cytoplasm, our mitochondria, and our nucleus. Now, when we look at the ultrastructure of the nucleus in the next couple of minutes, we will look at what the nucleolus, the nuclear pore, and the nuclear envelope actually are, but they're just all structures within the nucleus. Now, the first structure we're going to look at in detail is the cell membrane. So cell membranes are made up of phospholipids and proteins, so 2 structures, and we can see them here. So these are our phospholipids. It's a phospholipid bilayer. The reason it has that term is because we can see that we have 1 and 2 phospholipids on top of each other, and they're attached to proteins here in the center. So the phospholipids and the proteins are in constant motion, and the membranes are said to be fluid. Now, what are the functions of the cell membrane? Well, it's to separate the cell organelles and the cytoplasm from the outside, okay? It controls what gets in and what gets out. The cell membrane is referred to as being semi-permeable, so it allows some molecules freely in and out and others to enter. Membranes give some support to the cell, and the membranes recognize molecules that touch them, which is really important as well. Now, the next structure we have is our nucleus. So the nucleus is a large organelle near the center of the cell. It contains the cell's genetic information. There is other genetic information found in the cell in very small amounts, but most of the genetic information is going to be found within the nucleus, and it controls the activities of the cell. Okay, so it's the control center of the cell. Now, this is the ultrastructure of the nucleus, and some of these terms you would have seen a couple of minutes ago when we were looking at the overview of the animal cell. So this is our nucleus, and we can see we have our nuclear envelope up here towards the top, and that's for protection on the outside. So we have an outer nuclear membrane, and then we have an inner nuclear membrane as well. Nuclear pores are just holes that exist on the nucleus structure. We have our chromatin, and then we have our nucleolus in here towards the center. And the nucleolus is where ribosome formation will begin. And we can see these ribosomes down here, and they're hugely important in protein synthesis, which we have seen in a previous Saturday session. Okay, so that's the ultrastructure of the nucleus. We have our nuclear pore, we have our nuclear envelope, and we have our nucleolus. Now, we've kind of seen this already, but the nucleus is made up of a double membrane with numerous nuclear pores. So it's those nuclear pores that control the movement of substances into and out of the nucleus. A nucleolus contains RNA, DNA, and proteins, and that, as we said, was responsible for making the ribosomes. And then the chromatin, which contains DNA that is arranged into chromosomes, and that stores our genes. Okay, so that's responsible for inheritance. The mitochondria supply energy to the cell in a process known as respiration. Cells, therefore, with lots of mitochondria will produce a lot of energy, and cells with very few mitochondria will have very little energy. It's the inner membranes of the mitochondria that produce this energy. And just one other note there as well, we've mentioned so far about how the nucleus contains the genetic material. The mitochondria can contain small amounts of genetic material. And that is an exam question which has come up before. To give a non-nuclear location of DNA or genetic material, you can use the mitochondria as your answer for that. Now, we mentioned on the previous slide about how it's the inner foldings of the mitochondria that's responsible for the energy production. And we can see the inner membrane over here, okay? And the amount of energy that a mitochondria can produce is totally correlated to the amount of foldings that it has. So the more foldings that we can see here, the more energy it's going to be able to produce. So this mitochondria here is going to be able to produce a significant amount of energy. If we only had one or two folds the whole way along, that's going to be able to produce a lot less energy. Okay, so it's totally reliant on the inner membrane and the amount of foldings that it has. The cytoplasm then, this is a clear jelly-like fluid that fills the cell. Okay, that suspends all of the organelles. So it just makes sure that they're all spread out of each other and they literally just don't fall to the bottom of the cell. Okay, so all the organelles are suspended within the cytoplasm. It contains all of the organelles. And we can see here, both in animal and in plant cells, the vast majority of the inside of a cell can be taken up with the cytoplasm. Now, the plant cells have very similar structures to the animal cells. They also have a nucleus, a cell membrane, a vacuole, ribosomes, cytoplasm, and a mitochondria. They do have a couple of extra structures that we do not find in an animal cell. Okay, so that would be the chloroplast and the cell wall. One other difference that we would find there as well is that the vacuoles in animal cells would generally tend to be quite small, whereas when we move over to the plant cell, they would normally be quite large. Okay, so that would be another difference between animal and plant cells. Vacuoles in animal cells are small, they're larger in plant cells. But it's this cell wall and this chloroplast which we're really going to focus in on for the next couple of minutes because they're completely different from the animal cells. So what is the function of the chloroplast? Well, this is where photosynthesis occurs. So within the chloroplast, we have this green dye called chlorophyll. Okay, it's a green pigment. That's what absorbs the sunlight. And that sunlight can then be used to produce food for the plant, okay, in the process known as photosynthesis. So the chloroplast, their function is photosynthesis. Now, we said a couple of minutes ago about how the mitochondria can also contain some genetic material. The chloroplast can actually contain some genetic material there as well for the plant. Most of it, again, will be found in the nucleus, but some of it, if you were asked to give a non-nuclear location, the mitochondria or the chloroplast is an acceptable answer there. Now, the ultrastructure of the chloroplast, what would the chloroplast actually look like if we were to look at the inside structure? So the chlorophyll, the green dye which absorbs the sunlight, is found in these structures known as thylakoids. So you can see this kind of disc structure over here. It looks like there's a stack of CDs popped on top of each other. That's the thylakoids, and the chlorophyll is found on the inside of that. And it's that chlorophyll that will trap the sunlight energy, which can then be used for photosynthesis so that the plant can make its own food. Very like the structure of the nucleus, we can see we have an outer membrane and we have an inner membrane. So the thylakoids are these disc-like structures. The grana is the whole column of these disc-like structures, and the lumen is the inside. And then the final structure, which is found in plant cells, which is not found in animal cells, is the cell wall. So this exists outside the cell membrane. So it's the outer structure in a plant cell, and this is rigid and gives the plant cells a very defined shape. It's composed of cellulose fibers. So that's an example of a carbohydrate, a structural role of a carbohydrate. So polysaccharides and also some proteins, and the function of the cell wall is to support and to strengthen the cell. Now, we mentioned vacuoles already. These are found in animal cells. The only difference in vacuoles between the animal cells and the plant cells is that in animal cells, they tend to be quite small. In plant cells, they can tend to be quite large and sometimes can take up to 90% of the space of the inside of a cell. So they're membrane-bound sacs within the cytoplasm of a cell. They can provide structural support, but they also serve functions such as storage, waste disposal, protection, and growth as well. Plant cells, as we said, have large vacuoles. Now, in both of the examples we've had a look at so far in terms of the animal cell and the plant cell, both of them had a nucleus, okay? And they also had other membrane-bound organelles, such as the mitochondria or the chloroplast in the case of the plant cell. Because of that, that animal cell and the plant cell are known as eukaryotes, okay? Anything that's a eukaryote, like us, we will have cells that contain a nucleus and other membrane-bound organelles. Anything that's a prokaryote, an example of that being bacteria, they will not have a nucleus and they will not have other membrane-bound organelles. And that's a very, very common exam question to distinguish between eukaryotes and prokaryotes. You can just say eukaryotes contain a nucleus and other membrane-bound organelles, prokaryotes do not. An example of a eukaryote will be us, will be humans, or any other animal. Prokaryotes, an example of that, will be bacteria. Now, as part of this unit, you actually have two experiments to carry out in school. One of them is the preparation and examination of a stained animal cell, and the other is the preparation and examination of a stained plant cell. Now, the procedures for both are very, very similar. You're going to be using the microscope in a very similar way. The only difference is how did you source your animal cell? What was the animal cell you used? It's going to be completely different, obviously, from a plant cell. And then what stain did you use so that you could see the animal cell? And what stain did you use so you could see the plant cell? You sometimes will be asked to draw a quick diagram of what you actually seen in the microscope as well, but more often than not, it's going to be how did you prepare the slide? What stain did you use for the animal cell, or what stain did you use for the plant cell? Where did you get your animal cell? Where did you get your plant cell? Why did you add a cover slip? Things like that. That's the main questions that come up, and we'll see that in a couple of minutes when we look at an exam question. So the materials you need for the animal cell one was a microscope. You need slides, you need cover slips, and the stain that you use in this one is methyl blue or methylene blue. Okay, so that's why when you look at the animal cells underneath the microscope, they will appear in this kind of blue colour. It's because you've used a stain called methyl blue. Why do we add a stain? That's a question that comes up as well. So it's to see the animal cells more clearly. And it is really important that you know the name of that stain, because that question comes up all of the time. So another question that comes up is, what did you use as your animal cell, or how did you obtain your sample of animal cells? And that's what it says here. So I swabbed the inside cheek surface and transferred the sample onto a slide. Okay, so you got a cotton bud, you swabbed the inside of your cheek surface for a couple of seconds, and then you transferred the sample onto a slide. So just before that, one extra point you could put in there you normally would have put a drop of water onto the slide first, because that's going to make it's going to make sure that, first of all, the animal cells don't dry out and don't shrivel up, and it actually will make them swell up that little bit more on the slide as well to help make sure that you see them. It'll make them that little bit more visible. So once you've put the drop of water on your slide, you swab the inside of your cheek. You've then rubbed that cotton bud onto the water, and your sample or your cells will have moved over onto the slide. You're then going to put 1 drop of your stain to make sure that you can actually see the cells on top of the sample. So you put 1 drop of metal blue. You're going to allow it to stand, and then you're going to apply a cover slip. So that's another question that sometimes comes up as well, is how did you apply the cover slip? So you can say you applied it at a 45-degree angle using a mounted needle or tweezers, slowly, and the reason you did this is to prevent air bubbles. Okay, so then you examined it under the microscope, and I'm just going to add in one more point there as well, starting on the low power lens and then moving to high power. So you probably did a very similar experiment even at junior cycle level, but for this one, really important to note the difference between how or where you got your samples for both the animal cell and the plant cell, and then what stains you used. So for the animal cell, it's methyl blue. Now for the plant cell, a little bit different. We're obviously not going to be using cheek cells for this because that's an animal cell. You probably would have used something like an onion. Okay, so we're going to set up the microscope the exact same way. It says I cut the onion and located the epidermis. So the epidermis will be the outer layer. You cut the epidermis into small pieces and place them into water. Now, why did we place them into water? It's to swell the cells to make them more visible. So you transferred 1 piece onto the slide. Again, you're going to put a drop of water onto the slide first of all, just to make sure that it's not going to dry out. You applied a cover slip, but before you add the cover slip, you have to add your stain. And in this case, the stain is iodine solution. Okay, so it's kind of like a browny orange colour, and that's going to really cling to the carbohydrates that are in the cell walls and stain those, so it's more visible. They're really visible to see, and the exact same for the nucleus there as well. So as methyl blue for the animal cell, it's iodine for the plant cell. And the exact same thing is going to happen here. You're going to start to view the cells, starting with the low power lens on the microscope, working your way up to the high power lens. And you should see some differences straight away. In the last slide, we would have seen about the animal cells. They were more irregular shaped. We can tell straight away that these are plant cells up here because we can see that they're more regular shaped. They're kind of all the same size and same shape. And we can also see there the presence of the cell wall as well. So it is a little bit difficult to see, but you can probably see that there is a double layer in there, indicating that that's coming from a cell, a plant cell, and not an animal cell. So we're going to have a look at a quick exam question here then, based on everything we've seen so far. So distinguish clearly between a eukaryotic cell and a prokaryotic cell by writing a brief sentence on each. So a eukaryotic cell contains a nucleus and other membrane-bound organelles. And prokaryotic cells do not contain a nucleus and other membrane-bound organelles. So part B, a student observed the following images when examining stained cells using a light microscope. Image A was observed at 400 times and image B at 100 times magnification. Which image A or B represents the plant tissue? So we know that's going to be image B down here. Give a reason for this, a couple of different reasons you could do here. You could say that there's a cell wall present. You could have said that there's a more regular shape in B compared to A, whatever. Okay, just make sure you're only given one reason for your answer here, because if you were to give 2 reasons for your answer, one of them right and one of them wrong, the wrong one will cancel out the correct answer. So just one reason there. Identify structure Z, so that's the one here towards the center that contains the genetic material, so that's the nucleus. Part 4, when examining cells with a microscope, name a stain that can be used. So you can use a stain for either the animal cell or the plant cell in this case. It didn't actually specify. So I'm going to say methyl blue. That was the animal cell one. You could have said iodine there as well. Give one benefit of using a stain, makes the cells more visible. So the image of the cell in A was 2 centimeters wide. What is the actual width of this cell? Now in A, if we go back up here, we were told that this is magnifying it 400 times. And we were told that the image in A was 2 centimeters wide. So when we viewed it in the microscope, we could see that it was 2 centimeters wide, but that was after we magnified 400 times. So it was actually, we were viewing it 400 times bigger than it actually was. So a little bit of rough work over here at the side. If you divide 2 by 400, you will get 0.005. Okay, so it's 0.005 centimeters. Now you could actually left your answer as 2 over 400 centimeters there as well, or you could actually have given your answer as 5 by 10 to the power minus 3 if you want to use scientific notation. Any of those answers would have been accepted. And image B shows cells at 100 times magnification. Describe the steps taken to view these cells at 400 times. So you would move microscope. I'm going to have to do this over here at the side. So move to the higher power lens. We'd then use the coarse focus wheel. And then we would use the fine focus wheel. And that would have got you full marks there. So that was the structure of the cell, and for the next couple of minutes we're going to look at how those individual cells actually divide to produce more daughter cells. Okay, so cell continuity refers to how all cells develop from cells that already exist. And there were 2 types of cells which we're going to be mentioning over the next couple of minutes. We have haploid cells and we have diploid cells. Now before we get into the explanation of what each one of those is, each human cell will have 46 chromosomes. Okay, that's a full complement. Okay, we can't have any more, we can't have any less. There is specific cells, the sex cells being particular, the sperm and the egg, that will carry less, but the vast majority of cells will have 46 chromosomes. Okay, if you've any more or any less, it could lead to genetic disorders. So if you have a haploid cell, they will be ones that carry one set of chromosomes. So they will carry just 23 chromosomes, the sex cells, the sperm and the egg being examples of that. All other cells will have 2 sets of chromosomes, so we will refer to that as having 46 chromosomes. Okay, so haploid is 1 set of chromosomes, diploid is 2 sets. So it's either going to be N is 23, or then you would have 2 N's, which would be 46 for diploid. So chromosomes are coiled threads of DNA and protein. Okay, very common exam question actually to come up. What are the 2 things that chromosomes consist of? DNA and protein being the answer for that. Now we've already mentioned this already. This will be an example of a diploid cell down here, because we can see that we have 46 chromosomes or 23 pairs. Okay, all of the chromosomes are paired off in terms of their size and their shape. So you can see here that this first set of chromosomes here, they roughly have the same size and shape. If we go down to this one over here, the exact same thing. Now the first 22 pairs are numbered 1 to 22, but by the time you get to the 23rd pair, they actually aren't numbered. They just have either the letters X or Y. So if you have an X and an X, that individual will be female. If you have an X and a Y, they will be male. Now, where are these chromosomes actually located? Well, here we have our cell. Inside that cell, we have a nucleus, and inside that nucleus, we have our chromosome. Okay, we can see in this diagram down here as well that we have these bands that appear across these chromosomes. They're referred to as genes. Okay, and it's those genes that control inheritance and control inheritable characteristics like hair color, eye color, and so on. Okay, and they're just short sequences of DNA, but they do control all of the inheritable characteristics. So the cell cycle then, most cells will grow and then begin to divide. Cells have a life cycle known as the cell cycle, and there are 2 main phases within this cycle. The first phase, which can take up the majority of the cell cycle, is known as interphase, and then the second phase is where cell division actually occurs, and that's either going to be mitosis or meiosis, but we're going to be focusing on mitosis for the next couple of minutes. So interphase, this is the phase where the cell does not undergo cell division. It's the longest phase and can take up to 90% of the cell cycle. Now, it doesn't mean that nothing is actually happening. It just means that the cell is just not dividing. There is other important functions actually happening in interphase, which we'll get to in a couple of minutes. Now, in this phase, you actually cannot distinguish between individual chromosomes. So there will just be a mass of visible material located in here known as chromatin or chromatin. Okay, so this diagram is probably understating the fact here because you kind of can see some of the individual chromosomes, but if you can imagine, it's literally just going to look like a mass of material on the inside. That's known as the chromatin. Now, does anything actually happen with an interphase? And the answer is yes, it does. So the cell is not dividing, but it is producing new organelles such as mitochondria and chloroplasts, because eventually these are going to have to be passed off to the daughter cells. Okay, so organelles are being produced, and chromosomes are also beginning to make copies of themselves, because we want to make sure that sufficient genetic material is going from the parent cell down to the daughter cell. So not only are the new organelles being produced, but we do have some replication of genetic material happening within interphase as well. Now, after interphase, there are two types of cell division that can take place. We can have mitosis or we can have meiosis. Now, remember, in human cells, we only ever want to have a maximum of 46 chromosomes. If you have any more or any less, that can lead to complications. So at the end, we want to have 46 in the full entire organism. Now, mitosis, we're going to be dealing with cells that start off with 46 chromosomes and end up with 46 chromosomes. I know meiosis looks a little bit different because we're starting off with 46 and ending up with cells that have 23 and 23, but they are actually the sex chromosomes. They're supposed to have, or sex cells, they're supposed to have 23 chromosomes because the sperm at 23 will fuse with the egg, which is 23, and that combined will give 46. So they're only supposed to have 23. Now, we're mostly going to be focusing on mitosis and the 4 stages of mitosis today, but we will have a brief look at meiosis and where that actually occurs as well. So once interphase, which can be 90% of the cell cycle, is finished, cell division is going to occur. So mitosis is a form of nuclear division in which one nucleus divides to form 2 nuclei or 2 daughter cells, each one having the exact same number of chromosomes and identical genes. Okay, so they're exact copies. Whatever you end up with up here, or start off with up here, you end up with down here. And we can see that in this diagram over here. So here's mitosis. We have one cell here. We end up with 2 different daughter cells. Each one has an exact copy of what it originally had. Okay, so we can see here that it started to separate. We did have 2 of the red copies, which is exactly what we have down here, and we 2 of the blue copies, which is exactly what we have down here as well. So they're identical cells in mitosis. We go from 1 to 2, 46 chromosomes to 46 chromosomes. Now, there are 4 stages for this to happen to get from our parent cell down to our 2 daughter cells. We've prophase, we've metaphase, we've anaphase, and we've telophase. So in prophase, the chromosomes are going to begin to contract and become visible, because remember, within interphase, they were literally just existing as a visible mass of material known as chromatin. So they're going to start to contract and become visible. Each chromosome is now going to appear as a duplicated strand. So we have 2 chromosomes attached to each other here. Fibres are going to begin to form in the cytoplasm. So these are the spindle fibres, which we have coming up here, coming out of the centrosome. And this space here in the center has a very similar name, actually. That's referred to as the centromere. So the place where the 2 chromosomes are joined is referred to as the centromere. And the nuclear membrane here is also going to start to break down. Okay, and we can see that here. It's not a thick line or a solid line that's been used to annotate the nucleus. It's a dotted line showing that it's starting to break down. So that's prophase. Now, by the time we get to metaphase, the nuclear membrane has been completely broken down. The fibres have moved from the centrosome here, and they've now attached to the centromere from both sides. Okay, so from this centrosome and this centrosome, spindle fibres are attaching to the centromere from both sides. Not only that, you can tell that this is metaphase straight away because the chromosomes have started to line up at the centre of the cell or at the equator of the cell. Okay, so they're all going to be lined up in the exact centre down here. So we've gone from prophase, where the nuclear membrane is breaking down and we could visibly see the chromosomes, to where the nucleus is completely broken down. The chromosomes are towards the centre as a duplicated pair and spindle fibres are attaching to them. So for anaphase then, the fibres are going to contract and chromosomes are going to be pulled apart. So at the centromere where they were joined, that's going to be split, and the 2 strands are going to be pulled to opposite ends of the cell. So we can see here, they were existing down here towards the centre. One strand is going up here to the top half, the other strand is going down here to the bottom half. And then the final step is telophase. So this is where a nuclear membrane will form around each of the 2 sets of chromosomes and they will begin to condense into chromatin. Now at this point, the cells haven't actually fully split from one cell into 2 daughter cells, but it is beginning to happen. I can actually tell straight away over here that this is an example of an animal cell because of the way that the cell is beginning to split. This is called a cleavage furrow. So that's what happens in animal cells for them to split. So it kind of looks like a peanut shape there. It's pinching in towards the center, and eventually that will completely pinch in and the two cells will break off, going from one parent cell to two daughter cells. But depending on whether it's an animal cell or a plant cell, that splitting from going from the original cell to two daughter cells is completely different, which we will get into on the next slide. So the final stage, once we've prophase, metaphase, anaphase and telophase done, that is the 4 stages of mitosis, it still does need to split into the two daughter cells. And this is referred to as cytokinesis. So in animal cells, we've actually already seen this already. We form what's called a cleavage furrow. So it starts to pinch in and pinch in and pinch in, and eventually that one cell will split into two cells. Whereas over in the plant cell, we form what's called a cell plate. So this is just an abundance of vesicles or vesicles that will form up here, and eventually that's going to form the new cell membrane and cell wall, and that will split it from one cell into two different plant cells. Now, it is important to know the difference between both of those. The ins and outs of the processes of what's actually happening within the cleavage furrow and what's actually happening within the cell plate isn't as important. The question that most often comes up for this is, how is it different in the splitting of the cells in animal cells and plant cells, or how does it differ from one to the other? You just have to say that animal cells use a cleavage furrow, plant cells use cell plates. That's as far as you need to know for that. Now, why do we actually need mitosis? Well, unicellular organisms, now we're not unicellular, we're multicellular, but unicellular organisms use it to increase their numbers. That's how they actually produce. Us as multicellular organisms, we use it for growth and repair, and plants can also use it for growth and repair as well. Now, the other type of cell division after interphase which can occur is meiosis. So meiosis is a form of nuclear division in which 4 daughter cells, but they will only contain half the number of chromosomes as the parent. Meiosis generally occurs in the ovaries and the testes to produce sex cells, known as gametes, and this is why we get 23 chromosomes from each parent, because remember, we want the full cell to have 46. We couldn't have 46 being passed off from one parent and 46 being passed off from another parent, because then we'd end up with a genetic disorder. Okay, we have to have 23 and 23, which will combine to give us the 46. So it's a little bit different from mitosis. We do have one parent cell in both, but in mitosis, we'll end up with 2 daughter cells. Both of them will have 46 chromosomes. In meiosis, we have 4 daughter cells. Each of them will have 23 chromosomes. So that will be a difference. If you're asked that as part of an exam question, to give a difference between mitosis and meiosis, you can say meiosis has 4 daughter cells, mitosis has 2. Meiosis have 23 chromosomes in each of the daughter cells. Mitosis has 46 in each of the daughter cells. Okay, so we're going to have a look at a couple of exam questions then based on cell division. So the diagram shows a nucleus of a diploid cell undergoing one stage of mitosis. Answer the following in relation to mitosis. So the first one, we have to name the stage of mitosis shown in the diagram. So we know that's either going to be prophase, metaphase, anaphase, or telophase. Now, in prophase, the nuclear membrane starts to divide. We can individually see the chromosomes now. In metaphase, the nuclear membrane is broken down and they're lined up towards the center. And then in anaphase, they start to pull from one side of the cell to the other. And that's exactly what we can see here. So this is anaphase. Now it says justify your answer at part A above. How do we know that this is actually anaphase? A couple of different answers you could say here. You could say the spindle is contracting. And pulling chromosomes to one end of the cell. Identify the part labelled X in the diagram. That's the spindle fibre. Explain the term diploid. So we had the terms haploid and diploid earlier on. Haploid was containing one set of chromosomes. Diploid was containing 2 sets of chromosomes. What is the diploid number of this cell? So that's 4. Outline one function of mitosis in humans. So why do our cells need to undergo mitosis? You can say growth or repair. I'll have to do this question over here at the side. Outline one example of a human cell not produced by mitosis. So there you can say either the sex cell or the egg cell, either or. For a question like that, it did say give one example for both of those last 2 questions there actually. So again, just make sure you're giving one answer only, because if you give 2 answers and the second one is wrong, the wrong one again will cancel out the correct answer. Okay, so that's one exam question. Second exam question here, the photograph below is of plant cells undergoing different stages of mitosis. The cells are not shown in any particular order. Which type of microscope is used in the school lab to observe cells like these? So we've seen that earlier on, that was the light microscope. The first cell is in prophase. Give 2 observable events that happen during prophase. So nuclear membrane starts to break down. Different options then for the second one. So you can say the spindle starts to form. You can say the chromosomes become visible, or you can say that they become shorter, or you can say that actually they exist as double-stranded, okay, attached together by the centromere. On the photograph above, use the letter M to label a cell that is undergoing metaphase. So we prophase, metaphase. Okay, so for metaphase, they're going to be lined up towards the center. I can see that they're lined up towards the center here in this one. So I'll put M here. On the photograph, use letter A to label a cell that is undergoing anaphase. So anaphase is after metaphase, they're being pulled from one end to another. So I'll put the letter A here on the second one. And after mitosis, the cell divides in 2. This happens differently in animal cells and plant cells. Describe what happens in animal cells and in plant cells during this stage. So in animal cells, we form a cleavage furrow. You literally just have to mention those words. And in plant cells, we form our cell plate. Again, that's enough for full marks in that question. Okay, so 2 exam questions based on the cell cycle. They're very repetitive questions. It's generally the same things that will pop up there. Very, very manageable questions. So just before we finish up, I just want to take you through the ExamRevision website. So when you log on to ExamRevision, you can see that we have a wide range of subjects to choose from, not just biology, but all of them are going to be laid out the exact same way. So we have our video tutorials, we have our presentations, we have our exam builder, we have quizzes, and we have our H1 notes as well. So if you wanted to access the content from today's session, it's in Unit 2. It's in Unit 2.1, which is cell structure. So there's a video on cell structure and microscopy. And then we're also going to be in cell continuity, which is 2.3. And that video there will talk you through haploid, diploid, and the different stages of mitosis, and then give an overview of meiosis as well. Now, all topics are laid out the exact same way. There is video tutorials for you to click into. You can watch them at your own pace and watch them again and again and again until you're fully confident in all of the material. Once you're happy with the video, you can go back. You'll see here that we have MCQ quizzes, which you can try. So this one here on cell structure has 8 questions. You can submit your answers, you'll get your score straight away, and you can do the quiz again and again and again until you're comfortable and you're getting 100%. Once you're happy with the quiz, you can go down a little bit further. We have our exam builder. So not only can you look at the questions here based on the cell structure, so I'm going to add that one to my exam. You can go back up and you can choose questions from any topic that you wish, okay? So over here then, you can download your questions, you can download your answers, and test your knowledge on this topic and many other topics as well. If I just go back here quickly as well, we do also have our set of H1 notes. So you can click into these, you can download them, you can print them, and you can use them then to help you answer the questions and help you with your study. Now, there is the option to sign up for a year. The more subjects you do sign up for, the cheaper it does become for your 2nd, 3rd, and 4th subject and so on. But you can also sign up on a monthly membership and you can cancel it at any time as well. So it's a really, really useful platform for you to get access to H1 notes, video tutorials and quizzes based on not just the Leaving Cert Biology curriculum, but all subjects for the Leaving Cert. We do also run revision courses throughout the year, so do keep an eye on our social media channels for details about those that will be popping up throughout the year, and we will have lots more Saturday sessions popping up over the next couple of weeks as well. So thanks again for joining us today, and I hope to see you at a Saturday session for biology again soon.